Good evening, everyone, and welcome to this webinar on bioinformatics, a must for every biologist. I am Kashmila, and I will be the host for today's webinar. I am thrilled to see such a diverse and enthusiastic global audience who have joined us today for this webinar. In the realm of biology and technology, bioinformatics serves as a crucial bridge. Today, we are fortunate to have two of the distinguished speakers, Dr. Pratibha Singh and Mr. Rajavarman Kittu. Let me introduce you all to our eminent speakers. Dr. Pratibha Singh, as our opening speaker today, she is a visionary leader and innovator in the field of biosciences. Her extensive background in cutting edge research and her unwavering dedication to driving positive change have truly shaped the landscape of biotechnology. With her passion and expertise, Dr. Pratibha spearheaded the founding of Avanik Biosciences, a company that is truly at the forefront of pushing the boundaries of scientific discovery. But that's not all. We are privileged to also have Mr. Rajavarman Kittu, who is a clinical bioinformatics specialist with over 6 years of groundbreaking contribution. His journey has paved the way of aspiring professionals. His, aspire, his expertise has merged biology and technology, shaping data driven healthcare solutions. He has made significant strides at Kokilaben Dhirubhai Ambani Hospital and Primas Life Science Private Limited. His insights today are a result of years of commitment to advancing bioinformatics. So, without further delay, let's welcome Dr. Pratibha Singh and Mr. Rajavarman Kittur, prepared Prepare to expand your horizons at the crossroads of biology and technology. Thank you all for joining us, and now we shall begin with our webinar sessions. I request Dr. Pratibha to take over the platform and give us an insightful ride.
Thank you for that introduction, Kashmila. Good evening, everybody. Can you all hear me fine?
Yes, ma'am.
Yeah.
All right.
Yes.
Well, Kashmila has already, I think, introduced me well enough, so I don't really need to say anything more about myself. I think I will get directly into my presentation, which is about bioinformatics for biologists. I am an out-and-out biologist. Most of my work has been in wet lab, but none of us can really do without bioinformatics these days. Whether or not we practice it, we all benefit from the insights it provides. I am going to switch off my camera at the moment when I start sharing my screen, and I will switch it on once I'm done. I'm hoping that's fine by everybody. So see you in a bit. Can you guys see my screen?
Yes, ma'am.
All right, I'm just putting it on presentation mode. And through this presentation today, I will try and answer 3 questions. What is bioinformatics? Why is it important? And what can we use it for? But you're seeing on the screen, you're seeing 2 pictures. One looks like a tablet. The other is obviously the chromosome. And probably you're wondering what that's all about. So let me get into that. We are, talking about bioinformatics today, and it is definitely the science of the future. But I just thought, we'll start with a little, trip down history. What we're seeing on the left is the Rosetta tablet. It is a tablet a group of French soldiers dug up back in 1799. And prior to that, plenty of other tablets had been dug up, and there was hieroglyphics on them. But because till that point of time, nobody had been able to decipher what the hieroglyphics really meant, not much was known about Egyptian civilization in terms of where it began, how it did, the kings, and all of that stuff. What we know about a lot of other civilizations. So what was really important about this particular tablet was that along with the hieroglyphics, there was 2 more scripts on it, the Demotic and the Ancient Greek. And that was actually quite helpful, because the ancient Greek was a literal translation of what the hieroglyphics were saying on the tablet. And scholars of classical languages who were familiar with the Greek language, Greek script in particular, could actually translate. And once these translations began coming out, became to be a bit more available to the community of historians and archaeologists, they could then put together, read the script. They could, find out more about the civilization. And this is the same thing that is now happening with biology, right? We face the same challenges that the historians faced. The DNA is our book of life, and the arrangement of the nucleotides, right? They encode our similarities, our differences among ourselves and among different species. They govern our health, they govern our disease status, and so much more. The bioinformatics here is like that Greek script, and the bioinformaticians are those scholars that are trained in the classical languages, and they can make sense out of the biological data that is, coming out really fast and furious for all of us. For example, we all know that in our cells, right, we have chromosomes like these. um, I don't think I can point on here, but we have these chromosomes, and for a human chromosome, let's, for example, there's 23 pairs of chromosomes, 3.3 billion nucleotide pairs, around 23 to 30,000 genes, and they are coding for approximately 500,000 proteins. Now, these genes by themselves or in combination with each other, they exert subtle effects, on the protein formation, on our different signaling pathways. And it's important to know what they are doing. Providing, and bioinformatics, and, so I'm sorry, understanding this functionality of the genes by themselves and in combination with different protein complexes, it's very important to how we understand personalized medicine and how it is practiced today. If I go down, despite the fact that we have sequences available, right? Just reading off those long stretches of ATC gene and figuring out which are the genes among them. How are they being controlled? How are they being expressed? That is something that the bioinformaticians can do for us. And it's impossible to do it by ourselves, because not its human genome got sequenced back in 2000. And since then, 165,000 species have been sequenced. 1,000 trillion base pairs have been sequenced by now. I mean, just reading all of this information, only reading, not trying to do anything else with it, will take 30 million years. Moving on, what we see here is the number of nucleotide pairs that have been deposited in the GenBank since 1982. And this information has been doubling every 20 months. It's huge and it's growing exponentially in size. And this is being made possible because earlier it was just Sanger sequencing. Now there's next generation sequencing, and practically one human genome can be sequenced in a day with a very reduced cost as compared to earlier. This high throughput data, is giving us a tremendous opportunity to understand life. And it is allowing us to study questions that we could not have imagined studying before. It is allowing us to answer questions that were impossible to answer before. It allows us to study the difference between people at genome scale and answer many questions in population genetics. It allows us to understand genetic differences between normal and diseased condition. And it allows us to identify mutations that are responsible for these diseases. And using high throughput studies, we can also, find out how we can actually cure these diseases or, better manage their symptoms.